Somewhere under the Arctic right now, a shark is moving through black water at the speed of a slow walk. It was already alive when the American colonies were still colonies, not its species. That animal. And for most of the last century, nobody could prove it because this shark's skeleton keeps no record of time. No rings, no bands, nothing to count. The answer came from something far stranger. Fallout from nuclear weapons tests, and a piece of tissue inside its eye that stopped living before the shark was ever born. That tissue had been waiting for us. Here is the problem with that claim, and you should feel it immediately because scientists felt it for decades. If you want to know how old an animal is, you need the animal to have written it down somewhere. Most of them do. A tree writes it in wood. Every year, the trunk lays down a ring, wide in good seasons, thin in bad ones, and if you cut across it, you are reading a calendar the tree built out of itself. Most fish do something similar in a much smaller space. Inside the skull of a cod or a haddock, there is a tiny stone called an otolith, a hard little pebble of calcium carbonate that grows in layers, and if you slice it and hold it under the right light, you can count the seasons like pages. It is one of the strangest gifts biology ever handed to science. The animal keeps its own record and never knows it. Sharks are harder, but they are not impossible, and for a long time we thought we had them figured out. A shark's skeleton is not bone. It is cartilage, the same flexible material in the tip of your nose and the rim of your ear. And cartilage on its own is a terrible archive, because it does not lock minerals in place the way bone does. But sharks cheat. In the vertebrae, the stacked discs that run down the spine and take the load every time the tail beats, sharks reinforce the cartilage with calcium salts. They mineralize it. And that mineralization does not happen evenly through the year. It lays down in alternating zones, denser and less dense. And when you cut a shark vertebra in half and polish the face, you get something that looks disturbingly like a tree. Rings. Pairs of light and dark. Countable. That is how we know a great white can push past 70 years. That is how we know a whale shark is not a fast-living animal. You take the vertebra, you section it, you count, you calibrate the count against other evidence, and you get a number with an error bar on it. So somebody did that with a Greenland shark, and nothing happened. Not a bad reading, not an ambiguous reading, nothing usable at all. Picture the cross-section, a pale, slightly translucent disc, soft enough that it does not behave like the vertebra of a mako or a porbeagle under the blade. Where you expect the ordered stack of light and dark bands radiating out from the center, you get a smear, faint, inconsistent. The kind of pattern where two people looking at the same slide will give you two different answers, and neither of them can defend it. The Greenland shark's vertebrae are poorly calcified. It never built the archive. Sit with that for a second, because it is easy to hear it as a small technical failure, and it is not. It means that the animal now called the longest-lived vertebrate on Earth was, until very recently, one of the only large animals in the ocean we had no way to age. We could measure it, we could weigh it, we could dissect it, map its organs, sequence pieces of it. We could not answer the simplest question a child would ask about it. How old is that one? And here is the first twist, the one that reframes everything that follows. The failure was not the shark's. It was ours. Our entire toolkit for reading time out of an animal depends on that animal building something hard and leaving it alone. Bone, enamel, shell, otolith, mineral. We are very good at reading animals that invest in structure. This is an animal that appears to have spent its whole evolutionary history refusing to invest in anything it did not absolutely need. Why would a shark skip the reinforcement? Because reinforcement is expensive, and because it may not need it. A stiff, heavily mineralized skeleton is what you build if you are going to accelerate hard turn hard, and slam into prey at speed. Every one of those actions loads the spine, and the spine has to be rigid enough to transfer that force into the water instead of absorbing it. The great white's vertebral column is a spring under enormous strain. It has to be dense. Now imagine an animal that never accelerates. An animal whose entire life is a slow, straight-line cruise through water a couple of degrees above freezing, that almost never sprints, that has a liver so large and so full of low-density oil that it barely has to fight gravity at all. It hangs in the water column close to neutral. The forces running through its spine are a fraction of what a fast shark deals with. So the calcium never gets laid down in the same quantity because there is nothing demanding it, which means the missing rings are not a quirk. They are a symptom. The very thing that erased this animal's biography is the same thing that let it live long enough to have one worth reading. Cheapness. A body that does not spend. Every gram of calcium a shark drags around and mineralizes into its own spine is energy. And this is an animal built around the principle that energy is the one thing it will not waste. It saved on the skeleton for the same reason it saved on everything else. You will see that pattern again. It is going to explain almost every strange thing about this animal, including the ones that sound like defects. But in the middle of the 20th century, none of that was an answer. It was just a dead end. And in the absence of a direct measurement, scientists did what scientists always do when the front door is locked. They went around the back. If you cannot read the animal's internal calendar, you can build an external one. Catch a shark, measure it, put a tag in it, let it go. Then wait, sometimes for years, and hope somebody catches it again. When they do, you measure it a second time, and the difference between those two lengths, divided by the years in between, gives you a growth rate. It is crude. It depends on luck and on fishermen bothering to report the tag. But it is real data, taken off a living animal, and it does not care whether the vertebrae cooperate. So they tagged Greenland sharks, and they waited. And when the recaptures came back, the numbers were so small, they were almost inside the measurement error. We are talking about growth on the order of 0.5 centimeter to 1 centimeter in a year. Take a ruler. Look at the gap between one small tick and the next. That is roughly what this animal adds to its entire body in the time it takes the Earth to go around the sun once. A cod can put on more length in a month. A tuna in a good season is a different animal by autumn. The Greenland shark is functionally the same animal it was last year, and the year before, and the decade before. Now run the arithmetic, because this is where the room goes quiet. These sharks reach 4 meters, 5 meters. There are credible reports of individuals over 6. Take a 4-meter animal, subtract the length it was born at. Pups come out somewhere around 35 to 45 centimeters, small enough to hold. And you have roughly 3.5 meters of growth to account for. At 1 centimeter a year, that is 3.5 centuries. At 0.5 centimeter a year, you are into numbers that do not sound like biology anymore. That should have been the end of it. Case closed, headline written. It was not, and the reason is important, because it is the difference between a fact and a guess that happens to be right. Growth rates are not constant. Almost no animal grows in a straight line. Most grow fast when they are small and slow down as they get large, which means that if you take a growth rate measured on adults and run it backward across the whole life of the animal, you will systematically overestimate its age, sometimes by a lot. The tagged sharks that came back were not a random sample either. They were the ones that got caught twice, in the places people fish, which is not the same as the population as a whole. And the measurement itself is nasty. You are trying to detect a few millimeters of change on an animal several meters long, hauled onto a wet deck, flexible, heavy, and not remotely interested in lying straight. So what scientists actually had was a strong suspicion and no way to test it. The Greenland shark might be centuries old. It might also be an animal that grows fast for its first 2 decades and then nearly stops, in which case, a 5-meter individual could be far younger than the arithmetic suggested. Both stories fit the evidence. That is not a comfortable place to be. That is a question sitting open for the better part of a century, with everybody quietly certain of the answer, and nobody able to prove it. To break it, you need something the shark cannot fake. You need a tissue that gets built once, gets sealed, and then never changes again for as long as the animal is alive. Not a tissue that grows slowly. A tissue that stops. Almost nothing in a living body does that. That is the whole point of being alive. Your skin turns over in weeks. The lining of your gut turns over in days. Even your bones, which feel like the permanent scaffolding you were issued at birth, are being demolished and rebuilt continuously by cells whose entire job is to tear down old mineral and lay down new. Most of the atoms in you are not the ones you started with. A body is not an object. It is a process that holds a shape, which is exactly why the record keeps getting overwritten. But there is one place in a vertebrate body where that process is not just paused, it is forbidden. And the reason it is forbidden is not preservation, or memory, or anything remotely to do with time. It is because that tissue has to be see-through. Hold a lens up to the light, any lens. The one in a camera, the one in a pair of glasses, the one you can lift out of a fisheye on a dissecting tray, a small clear sphere that looks like a marble made of jelly. Light has to pass through it and come out the other side bent to a point. That is the entire job. Now think about what that requirement forbids. It forbids blood vessels, because blood is opaque, and a network of capillaries running through a lens would throw shadows across everything you looked at. It forbids the ordinary furniture of a cell, the nucleus, the mitochondria, the internal membranes, because every one of those is a boundary between materials of different density, and every boundary scatters light. A normal cell is a fog. Stack a few million of them in front of your retina, and you have exactly what a cataract is. A lens that has stopped being a window. So the lens does something no other tissue in the body does. It kills its own cells while keeping them. The cells in a vertebrate lens, called fiber cells, are laid down in long ribbons. And then, as they mature, they dismantle themselves from the inside. They eject the nucleus. They break down their organelles. What is left is a thin, elongated sac, packed almost solid with a family of proteins called crystallins, arranged so precisely and so densely that light passes through the whole array without scattering. The tissue is not dead in the sense that it rots. It is dead in the sense that it has given up the machinery of maintenance in exchange for being invisible, and it has no blood supply. None. There is no delivery route into the center of a lens. Follow that logic all the way down because it leads somewhere strange. New fiber cells are added at the outside of the lens throughout life, layer over layer, like the coats of an onion. But the center, the nucleus of the lens, the innermost core, is laid down first. It is built during embryonic development, before the animal is born. And because nothing can get in to replace it, and no cellular machinery survives in there to remodel it, the proteins in that core stay exactly as they were assembled for the entire life of the animal. Those proteins are among the oldest continuously existing structures in any vertebrate body. Not old like a scar, old like a sealed room. Now ask where the raw material came from. Proteins are built out of carbon, and the carbon in that lens core came from the developing embryo, which got it from its mother, who got it from what she ate, which got it from the food web she was hunting in, which ultimately got it from the water and the surface ocean above her. Which means the chemical signature of the ocean at the moment that shark was an embryo is locked inside its eye. and it has been sitting there, chemically untouched, ever since. The animal has been carrying a sample of the world it was born into. That is the trick. That is what broke the problem open. Not a better microscope, not a better fossil, a recognition that this one specific tissue is a time capsule, and that we already had the tools to read it, because we had spent decades learning to read carbon. Here is how carbon tells time, and it is worth getting right because the popular version is wrong in a way that matters for this animal. Most carbon in the world is carbon-12, stable, boring, permanent. A tiny fraction is carbon-14, which is radioactive and unstable. Carbon-14 is made continuously in the upper atmosphere when cosmic rays smash into nitrogen. It gets swept into carbon dioxide, taken up by plants and plankton, and passed through every food web on Earth, which means every living thing carries a small, roughly predictable dose of it. When something dies, or when a tissue stops exchanging carbon with the world, the intake stops and the clock starts. Carbon-14 decays with a half-life of about 5,630 years. Count how much is left, and you can say how long ago that tissue closed. That is how you date a mammoth tusk, or a piece of charcoal from a fire pit. It is close to useless for a few centuries, because 5,630 years is a very long half-life, and a 300-year-old sample has lost only a sliver of its carbon-14. The signal is there, but it is buried in the noise of measurement error and in the fact that the amount of carbon-14 in the environment has never been perfectly constant. On the scale of a shark's life, ordinary radiocarbon dating gives you a smear, not a date, which is where the 20th century did something monstrous and entirely by accident, useful. Between the early 1950s and the early 1960s, nations detonated nuclear weapons in the open atmosphere, not underground, in the air and over the ocean and on islands. Those detonations flooded the upper atmosphere with neutrons, and neutrons hitting nitrogen make carbon-14 exactly the way cosmic rays do, only vastly faster. In roughly a decade, atmospheric carbon-14 spiked. It rose to nearly double its natural level. Then, in 1963, the major powers signed a treaty that ended atmospheric testing, and the spike began to fall as that excess carbon was absorbed into oceans and plants and soil. Picture the graph because it is one of the most recognizable shapes in modern science. A flat line running across the centuries, then a near vertical wall going up in the late 50s and early 60s, then a long, smooth decline down the far side, still falling today. That wall is a timestamp. It is printed into every living thing on the planet. Any tissue that formed before the mid-1950s has natural pre-bomb carbon in it. Any tissue that formed during or just after the spike carries a signature that is unmistakably elevated. It is not a subtle isotopic wobble you have to argue about. It is a cliff. If you find the cliff in a tissue, you have found the early 1960s in that animal's life to within a few years. So put the two ideas together, and you have the method. Take the eye lenses out of Greenland sharks that have been killed as bycatch. And it has to be bycatch, because this is not a procedure you can do on a swimming animal. Dissect down to the innermost core of the lens, the part built before birth. Isolate that tissue. Measure its radiocarbon. If the core shows the bomb spike, the shark was born after atmospheric testing started. It is younger than the atomic age. That is a hard ceiling on its age, and it is not a matter of interpretation. If the core shows no spike at all, the shark was already an embryo before the bombs went off. It is older than the atomic age, full stop. That is what a research team did, publishing the result in 2016 in Science, working with 28 female Greenland sharks ranging from 81 centimeters up to just over 5 meters. And the bomb signal showed up only in the smallest animals in the set. Roughly the sharks around 220 centimeters and under. Read that carefully because it is doing enormous work. It means a Greenland shark just over 2 meters long, an animal you would look at and call a large shark, an animal bigger than the person measuring it, is a juvenile that was born after humans started detonating nuclear weapons in the open air. And it means that every single individual in the study larger than that had no bomb carbon in its lens core at all. Every one of them was already alive, already formed, before 1960. You do not need a model for that part. You do not need a growth curve or a statistical assumption. The carbon is either elevated or it is not. Those big sharks were not elevated. That alone would have been a headline. But it also gave the team something better: a fixed point. If you know that a shark of a certain size was born in the early 1960s, you now have one calibrated anchor on the growth curve, and you can start fitting the rest of the animals to it, using the older, slower carbon signal in the larger lenses to estimate how far back each one goes. The numbers that came out are the reason you are watching this. The analysis put the lifespan of the species at a minimum of about 272 years, not a maximum. A floor. And the largest animal in the set, the one just over 5 meters, came out at an estimated 392 years, plus or minus 120. That error bar is not a footnote, and I am not going to bury it, because the messy answer here is better than the tidy one. Plus or minus 120 years means the real answer for that individual could be as low as 272 or as high as 512. It is a wide window. It is wide because you are extracting a date out of a decayed isotope signal in a tissue from an animal nobody can watch, calibrated against ocean carbon that is itself complicated. Seawater carries carbon that has been out of contact with the atmosphere for a long time, so marine samples read older than land samples unless you correct for it, and that correction is its own field of argument. So no, we do not have a confirmed oldest individual with a birthday. Anyone who tells you a specific shark was born in a specific year is selling you a precision the data does not contain. But look at what the uncertainty actually does here. It does not rescue the ordinary explanation. It cannot, because the bottom of that range, the most conservative, most skeptical, take every assumption in the least dramatic direction reading, still lands at 272 years. Take a shark alive in the water today and subtract 272 years. You land in the middle of the 18th century, before the Declaration of Independence. Before the country most of you are watching this from was an idea anyone had written down. The skeptical answer is still the impossible one. And there is something genuinely dark about how we got it. To age an animal that predates the United States, we needed a signal that only exists because that country and its rivals spent a decade detonating nuclear weapons into the open sky. The most reckless thing our species has ever done to the atmosphere left a mark in every living cell on the planet, and that mark turned out to be a ruler. The bombs are how we learned that the sharks were older than the bombs. There is one more piece of ugly symmetry in this, and it comes back later. The tissue that gave us the answer sits behind a cornea that, in this species, is very often covered by something else, something living. The eye that told us how old these animals are is in a large fraction of individuals, an eye that may barely work at all. That sounds like an accident of the story. It is not. But before it makes sense, you need to understand what kind of body is capable of running for 3 centuries in the first place. Because a long life is not a reward, and nothing about it is free. It is a bill this animal pays every second it is alive, in a currency most predators refuse to give up. Researchers have put data logging tags on free-swimming Greenland sharks, the kind of tag that records speed and body movement while the animal goes about its business and is later recovered. The results published in 2012 are some of the least dramatic numbers in marine biology and some of the most revealing. Cruising speed came out at about 1/3 of a meter per second. A person walking without hurrying moves at roughly 1.5 meters per second, so a shark that can be longer than a car heavier than 3 or 4 adults, moves through the water at somewhere around a quarter of your walking pace. If you were in the water beside it, which the cold alone would not let you do for long, but hold the image, you would not have to swim to keep up. You would have to actively hold yourself back. The tail beat frequency is worse or better, depending on what you find unsettling. Around 1 beat every 6 or 7 seconds. Watch a normal shark, and the tail is a constant metronome, a side-to-side rhythm you can count without thinking. Now slow that down until you are waiting for it. The tail sweeps, nothing. Still nothing. The shark glides, dark and enormous, and just as you start to wonder if it has stopped, the tail sweeps again. It is closer to the rhythm of breathing than to swimming. Those measurements made it the slowest swimming fish for its body size ever recorded. And the obvious reaction is that this is a defect. A crippled animal. Something that got left behind. That is exactly backward, and the cold is the reason. Greenland sharks live in water that runs from roughly 0 to 5 or 6 degrees Celsius, and in parts of the Arctic they are moving through water that sits just below freezing and stays liquid only because it is salt. They are ectotherms. They do not generate meaningful internal heat. Their body temperature is the water's temperature, and that means the chemistry of every muscle fiber in them is running at the water's temperature too. Chemistry does not care about your intentions. Enzymes work by physically colliding with the molecules they act on, and how fast those collisions happen depends on how much thermal energy is in the system. Take heat out and everything slows. The enzymes that release energy from food, the ones that cycle the calcium that triggers a muscle contraction, the ones that reset the fiber so it can contract again. A muscle in near-freezing water cannot generate force at the rate a warm muscle can. It is not a matter of trying harder. The molecular machinery is being run at a fraction of its speed. Some fish fight this. Tuna and lamnid sharks, great whites, makos, poor beagles, evolved heat exchange systems that trap the warmth their own swimming muscles produce and keep the core of the body well above the surrounding water. It is an expensive solution. It works. It is why a mako can accelerate in cold water like something out of a different physics. The Greenland shark did not go that way. It surrendered, and the payoff for surrendering is the entire story. An animal that does not fight the cold has a metabolic rate that collapses to a small fraction of what a warm-bodied predator needs. That means it needs less oxygen, less food, less of everything, per hour, per day, per year. And when you take the pace of an animal's internal chemistry and divide it by 4 or by 8, the calendar it lives on stretches to match. But cold alone is not the answer. And I want to kill that explanation now because it is the lazy one, and it does not survive contact with the Arctic. The Arctic is full of cold animals that die young. Polar cod live in the same water and are old at 10 or 15 years. Capelin, the small silver fish that half the North Atlantic food web is built on, live a handful of years and then spawn themselves to death. Krill in cold water do not last centuries. If temperature by itself bought you 300 years, the poles would be full of ancient animals, and they are not. They are full of short-lived ones packed into a brutal, seasonal boom and bust system. So cold is necessary, it is not sufficient. Something else is going on, and it takes several things stacking at once. Start with size. Large bodies lose heat and burn energy more slowly per kilogram than small ones. A mouse's heart runs at hundreds of beats a minute, an elephant's at around 30. Being big is already a brake on metabolic pace. Stack cold on top of that, and you have compounded the effect. Then add behavior. This is not a large, cold animal that spends its energy budget on activity anyway. It moves slower than you walk and beats its tail every 7 seconds. It has stripped its own energy expenditure down to something close to the floor of what a moving vertebrate can do. Even its skeleton, as we saw, refuses to invest in unnecessary mineral. And then there's the part that is genuinely unsettled. And I am going to flag it clearly because the honest version of this is more interesting than the confident one. There is a long-standing idea in biology, usually called the rate of living hypothesis, that says the faster you burn, the sooner you break. The logic is straightforward. Metabolism is chemistry. Chemistry generates reactive byproducts. Those byproducts damage proteins and membranes and DNA, and damage accumulates. Run the engine slower and the damage arrives more slowly. It is intuitive, and it explains a lot of comparisons across species. It also fails in enough cases that biologists no longer treat it as a law. Birds have high metabolic rates and outlive mammals of the same size. Naked mole rats do not behave the way their metabolism predicts. So if you hear someone say the Greenland shark lives for centuries simply because it is cold and slow, that is a hypothesis wearing the costume of an explanation, which is why the recent genetic work is worth watching, and worth watching carefully. Researchers have now assembled the Greenland shark genome. It is enormous, roughly twice the size of the human genome, one of the largest ever sequenced outside four-limbed animals. Early analyses reported something specific, an unusual expansion of genes involved in DNA repair, genes that appear as single copies in other sharks and as multiple copies in this one, along with a change in a region of the p53 protein, which is one of the central controllers of what a cell does when its DNA is damaged. That points toward a real mechanism, not just an animal running slowly, but an animal that is actively better at fixing itself. It is also new. The first version of that work appeared as a preprint before it went through peer review, and the conclusions are still being tested and argued over. Gene duplication is suggestive. It is not proof of function. Finding extra copies of repair genes in a long-lived animal is the kind of result that can hold up beautifully or turn out to be an artifact of a difficult genome assembly. Watch that space. Do not treat it as settled. There is a hint from a different direction, though, and it is a strange one. When researchers examined the eyes of Greenland sharks over a century old, they reported retinas that looked intact, without the degeneration you might expect from 100+ years of use. Whatever this animal is doing, it does not appear to simply be a young shark that has been left running for a very long time. Something is maintaining it. And once you know what to look for, you can read the whole strategy off the outside of the animal before you cut into anything. Start with the name it was given: Somniosus microcephalus, sleepy and small-headed. Whoever named it was describing exactly what they saw on a deck. A sluggish thing with a short, rounded, unimpressive snout on a very large body. Then look at the fins, and think about what a fin actually is. A fin is a control surface. It generates lift, or it resists roll, or it lets the animal turn without spinning out. And the size of a control surface is set by the forces it has to control. A fast pelagic shark carries big, stiff, wing-like pectoral fins and a tall dorsal because at speed the water is shoving hard and the animal needs authority over its own body. The same reason a fighter aircraft has surfaces a glider does not. Greenland shark fins are small. The dorsals are low and set well back. The pectorals are modest for an animal of that length. There is nothing there that would let it carve a hard turn because it never makes one. It is not under-equipped. It is equipped for the forces it actually experiences, which are almost none. Then, look at the gill slits, which are the detail almost nobody notices and the one that gives the whole thing away. They are small. Notably small for the size of the animal. And gill area is not a stylistic choice. It is a direct physical measure of how much oxygen an animal needs to move across a membrane per minute. Tuna and mako sharks have enormous gill surface area, so large that they must swim continuously with their mouths open just to push enough water over it, because their oxygen demand never stops. That is what a high metabolic rate looks like from the outside. Small gills mean small demand. There is no way around it. You cannot run a hot engine through a narrow intake. So the metabolic story we just worked out from tags and temperature is written on the side of the animal's head in plain view, for anyone who knows what gill area means. The same logic runs inward. A heart in near freezing water is a slow heart because cardiac muscle is ectothermic tissue like everything else. The contraction cycle depends on the same temperature-limited chemistry. Recent work has begun examining whether the Greenland shark's heart shows the deterioration you would expect after a century or more of use, which is exactly the right question to ask about an animal like this, and that work is still at the preprint stage. Do not take conclusions from it yet. Note that somebody is finally asking, and digestion runs on the same clock as everything else. A cold-bodied animal with a low metabolic rate processes a meal slowly, which sounds like a disadvantage until you flip it around. A slow digestive system means a single large meal lasts an extremely long time, and an animal that can go months between feeding events without distress can afford a strategy that a fast metabolism cannot, waiting for something to happen instead of going out and forcing it. Each of these compounds the others. Low demand permits small gills. Small gills mean less tissue to build and perfuse. Less activity means less muscle to feed. A lighter skeleton to carry. Smaller control surfaces to drag through the water. And every one of those savings lowers the demand again. It is a body engineered downward, over and over, until almost nothing is left to spend. Now hold both halves of this in your head at once, because they are in tension. An animal that cannot generate force quickly. An animal that will not spend energy. An animal built entirely around endurance, rather than performance. That is a magnificent design for not dying. It is a catastrophic design for catching anything. Because energy is not just what keeps you alive between meals. Energy is what you spend to get the next meal. Predation, almost everywhere in the ocean, is a burst. It is a lunge, a strike, a snap of the jaws faster than the prey's nervous system can process. That burst is the single most expensive thing a predator ever does, and the Greenland shark appears to have opted out of the entire category. By every reasonable expectation, this should be a scavenger, a garbage disposal, an animal that drifts along the seafloor at 1/3 of a meter per second, following the smell of things that are already dead. Whale falls, discarded fish guts, drowned carcasses sinking out of the world above. That would be a perfectly respectable way to live, and it would fit the body perfectly. For a long time, that was the assumption. Then people started opening them up. Stomach content studies on Greenland sharks, Nimitariem, including work on animals sampled around Svalbard, turn up what you would expect from a slow, deep-water predator, fish, squid, the kind of material that sinks or swims badly. They also turn up seals, not occasionally. Seal remains show up often enough that they are treated as a real component of the diet, rather than a curiosity. And the sharks in question are animals that move slower than you walk. A ringed seal is not slow. It is a warm-blooded, streamlined, highly alert diving mammal that can outswim this shark without any meaningful effort, in a straight line, in a circle, while thinking about something else. There is no version of a chase where the shark wins. There is no version where the shark even gets to start one. So how is seal ending up inside it? There are two answers, and the argument between them has not been settled, which is the honest and much more interesting situation. The first answer is scavenging, and nobody disputes that it happens. The Arctic kills a lot of animals. Seals die of disease, injury, old age, and polar bears. And bears do not eat the whole animal. Carcasses sink. Fishing vessels dump offal. Whales die and go down as multi-ton parcels of food that can feed a deep-sea community for years. If you are a large animal with a low energy budget and a nose, the seafloor is a slow, reliable buffet, and getting there first requires patience rather than speed. A shark that burns almost nothing can afford to spend weeks going nowhere in particular, waiting for the smell of something dead to reach it. That is a legitimate way to make a living, and it almost certainly accounts for a great deal of what ends up in these stomachs. The problem is that scavenging does not explain everything found in them, because the remains are not always the picked-over leftovers you would expect from something that has been on the bottom for a while. Some of it looks fresh. Some of it is the kind of tissue that suggests the shark got there very early or first. And once you consider that, the whole question flips. You stop asking how a slow animal catches a fast one, and you start asking a much better question. How much speed do you actually need? Look at where the strike happens. Almost every predator we picture in the ocean closes distance with the body. The mako accelerates. The seal lunges. The whole animal is the weapon, and speed is how the weapon reaches the target. That is why the burst matters. That is what costs so much. But the actual killing is not done by the body. It is done by the mouth. And a mouth is a small, light structure, operating on short levers over a distance of a few centimeters. The speed of a jaw is not governed by the speed of the animal it is attached to, which means a slow shark can still have a fast bite. Greenland sharks are suction feeders, and suction feeding is one of the most underrated mechanisms in the ocean. The shark drops the floor of its mouth and expands the throat cavity, and because water cannot be compressed and does not tolerate a vacuum, the surrounding water rushes in to fill the space, along with whatever is floating in it. Done properly, prey is not chased, it is pulled. The animal does not have to intercept the target. It has to get close enough for the pressure gradient to do the work. Then there are the teeth. And this is where the design stops being subtle. The upper jaw carries narrow, pointed teeth, spikes. They are not built for cutting, and they are not built for holding on the way a great whites are. They are built to sink in and anchor. The lower jaw is a different tool entirely. The lower teeth are broad and squat, each with the cusp angled sharply to one side, and they interlock edge to edge along the whole span of the jaw. What that produces is not a row of individual teeth. It is one continuous blade running across the front of the animal's face. Now put the two together, because they are a single mechanism. The upper spikes drive in and hold. The shark rotates, a slow, heavy roll of the whole body. And the lower blade sweeps through the anchored tissue in a circular arc. It does not bite a piece off. It cuts a plug out. The result is a clean, round crater in the flesh of whatever it grabbed. You can see the same signature at miniature scale from the cookie cutter shark, a small deep-water species that takes coin-sized divots out of whales and tuna and leaves them swimming. Scale that concept up to an animal several meters long, and the plug is not a divot. It is a wound that removes a serious portion of an animal. Every part of that is a low-speed weapon. It requires no chase because it is a contact mechanism. It requires no pursuit after the strike because the shark is not trying to keep hold of a struggling seal. It is taking a section and letting the rest go. It does not even require the shark to be strong in the way we usually mean. It requires the shark to be there, and to be within a body length without the seal knowing. Which brings us to the part that genuinely changes how this animal feels. We treat speed as the frightening quality in a predator. It is not. Speed is loud. Everything that moves fast through water pushes a pressure wave in front of it and drags a turbulent trail behind it. That is unavoidable physics. And the ocean is full of animals whose sensory systems exist specifically to detect exactly that. Fish have lateral lines, rows of tiny sensors down the flanks that read pressure changes in the surrounding water. Seals have vibrissae. Those thick whiskers are not decoration. Experiments with harbor seals have shown they can follow the hydrodynamic trail left behind by a moving object, tracking a wake through water the way a dog follows a scent through grass, without seeing anything at all. Now consider what a Greenland shark presents to that system. A body moving at 1/3 of a meter per second, a tail that sweeps once every several seconds, no burst, no acceleration, no pressure wall shoved ahead of it. It has no swim bladder to resonate, and it is close to neutrally buoyant because of an enormous, oil-filled liver, so it is not thrashing to hold depth. It is running colder than the water around a warm-blooded mammal, in darkness, with almost no wake. To a sensory system built to notice movement, an animal this slow is close to nothing at all. That is the reversal. The slowness is not the reason it cannot hunt. Under a specific set of conditions, the slowness is the hunt. It is the only way to approach an alert marine mammal without triggering the alarm that speed would set off half a second too early. And there is one more condition that makes it work, and it is the one people find hardest to sit with. Seals sleep in the water. They have to. A diving mammal in the Arctic cannot spend its whole life hauled out on ice, and many pinnipeds rest floating at the surface or hanging in the water column, in a state where their attention is not where it should be. They do it in daylight and in the long polar dark. They do it in a place where the thing beneath them makes no noise and casts no wake and does not need to hurry. The scenario writes itself, and the biomechanics support it. A slow, silent, cold-bodied animal drifting up from below into an animal that is not paying attention, close enough for suction, one anchoring bite, one roll. And it has to happen shallow, which is the part that surprises people who have filed this animal under deep sea. Seals do not live at 2,000 meters. Whatever is happening between these two species is happening in the top of the water column, near ice and near coasts. And the Greenland shark is entirely capable of being there. Its recorded depth range starts at the surface, not near it. At it. These sharks have been found in shallow coastal water in the Arctic and around Iceland, and researchers have tracked them beneath sea ice, which quietly overturns something we have been assuming for the last few minutes. This is not a deep-sea shark that occasionally comes up. It is a cold water shark that can be anywhere cold. In most of the world, the only cold water is deep, so that is where it lives. In the high Arctic, the cold comes all the way to the top, and so does the shark. The depth range is not a preference. It is a consequence of a temperature limit. Now put that in an Arctic winter and think about what the geometry looks like. Under the ice, the world is dark, and the seals are still there because they have to be. They maintain breathing holes. They rest at the ice water boundary. They haul out and slip back in. And the threat their entire behavioral risk model is built around is a polar bear waiting at a hole. That danger comes from above and from the ice, and it announces itself with weight and noise and the smell of an enormous warm animal. A shark drifting up from below at 1/4 of walking speed in black water, generating almost no wake, is arriving from the one direction that model does not cover. That is reasoning, not observation, and I am labeling it as such. But it is the reasoning the biomechanics point toward, and it explains something the scavenging hypothesis alone struggles with. Why fresh marine mammal tissue keeps appearing inside an animal that should be too slow to get any. The frustrating part is that the same environment generates the alternative explanation just as neatly. Polar bears kill seals on the ice constantly and do not eat the whole animal. They favor the fat and often leave the rest. That material ends up in the water. So the exact setting where a shark could plausibly ambush a resting seal is also the setting most reliably littered with seal remains that something else killed. Both stories are produced by the same place. The evidence in the stomach cannot tell them apart. I want to be precise about what we actually know here, because this is where documentaries tend to lie. We do not have a confirmed, direct observation of a Greenland shark killing a healthy adult seal in the wild. Stomach contents on their own cannot tell you the difference between something that was hunted and something that was found. And fresh material can be explained by a carcass that hit the bottom recently. Both explanations survive the evidence. The most defensible position is the boring one. Some of it is scavenged, some of it very likely is not. And the balance between the two is unknown. That uncertainty is not a weakness in the story. It is the story. There is a several-meter predator moving through the Arctic in the dark that may be routinely killing marine mammals, and we cannot say for sure because nobody has been in the right place, at the right depth, at the right moment with a camera. And then there is the material that nobody has a comfortable explanation for. Reindeer have been reported from Greenland shark stomachs, so have remains attributed to polar bear. Horse has come up. Every one of those animals lives on land. Now there are reasonable explanations, and they should come first. Reindeer swim between islands and across straits and sometimes drown doing it. Carcasses wash into the sea from shorelines and rivers. Waste from settlements and hunting camps goes into the water. A shark that eats anything dead and sinking will inevitably eat things that were never marine. That is not mysterious. But there are also accounts from biologists raising the possibility that these sharks may take swimming land mammals opportunistically, the way a crocodile takes something crossing a river. That has not been documented in a way that would settle it, and it should be filed as a possibility rather than a fact. Keep the image anyway, because it is the correct one either way. Whatever the mechanism, the record inside these animals says that the boundary between the Arctic land and the Arctic Sea is not a wall. Things fall in, and something enormous and unhurried is down there, several hundred years into its life collecting them. Which leaves the question that should have been bothering you for the last few minutes. All of this. The ambush, the suction, the plug of flesh, the smell of a carcass 2 kilometers away requires the shark to find the target in the first place. And a very large proportion of these animals appear to be looking at the world through something that should not be there. Get close to the head of a Greenland shark, and the first thing you notice is not the mouth. It is that there is something hanging out of the eye, a pale, fleshy tube, a few centimeters long, trailing loose in the current with 2 thin strings streaming behind it. It moves when the water moves. It is unmistakably alive. And it is unmistakably not part of the shark. It is a copepod, a crustacean, distantly related to the shrimp in a supermarket freezer, from a group whose free-swimming relatives are among the most abundant animals on the planet. This particular species has abandoned all of that. The adult female finds a Greenland shark, settles on the surface of the eye, and stays. And it does not settle lightly. It anchors. The parasite drives a hold-fast structure down into the corneal tissue and locks itself in place, so that the part you can see waving in the water is only the exposed body of an animal that has physically rooted itself into the shark's field of vision. Then, it feeds on the surface of the eye it is standing on. The rate at which this happens is the part that stops people. In some surveyed populations, essentially every individual examined has carried at least one, not a majority. Not a common finding, close to all of them. The damage is documented. Around each anchoring point, there is an opaque patch where the cornea has been physically disrupted. There is erosion and scarring from the feeding. Where multiple parasites occupy the same eye, they can cover a substantial part of the corneal surface, and the tissue underneath is not a clear window anymore. It is a scratched, clouded, partly obstructed pane. So the picture you now have is an apex-sized Arctic predator, 3 centuries old, moving through black water with living animals bolted onto both eyes, seeing, at best, a blur. And the natural conclusion is that this is a tragedy. A slow, half-blind, parasitized wreck of a shark, surviving on garbage because nothing else is available to it. That conclusion is where almost everyone gets this animal wrong. Start with the environment, because the environment is what decides whether an eye is worth anything. Sunlight does not go far into seawater. In the clearest ocean, the last usable trace of it is gone somewhere below a few hundred meters. And Greenland sharks spend large amounts of time far below that, recorded down past 2,000 meters, and reported deeper still. Down there is no daylight. Not dim daylight. None. And even up shallow. This is the Arctic. For months at a time, the sun does not clear the horizon. Add a lid of sea ice over the top, and you have a surface layer that is, for a large part of the year, functionally as dark as the deep. Now ask what a scratched cornea costs an animal in a place where there is nothing to look at. That is the reversal. The parasite is not necessarily crippling this shark because clear vision may not be a sense this shark depends on for the things that keep it alive. You cannot be robbed of something you are not using. A corneal injury that would end a hunting bird is, in this environment, closer to a chronic irritation than a catastrophe. Except that answer is too tidy. And recent work has made it a lot more complicated. Because if vision were genuinely useless to this animal, evolution should have let the eye degrade. That is what usually happens. Cave fish lose their eyes. Deep burrowing mammals, Tabor male, reduce them to nubs under skin. Eyes are metabolically costly tissue, and a species that stops using one stops paying for it. The Greenland shark did not stop paying. Researchers examining the visual system of these sharks found an eye that is not neglected at all. It is specialized. The retina is dominated by rods, the photoreceptors built for low light, not the cones you use for color and daylight detail. And those rods are elongated and densely packed, which is the standard architecture for wringing information out of almost no photons. The retinal tissue is extremely rich in the long-chain fatty acids that keep photoreceptor membranes flexible and functional at proportions higher than in mammals. And the light-sensitive pigment itself has been shifted. The rhodopsin in a Greenland shark's rods was measured with a peak sensitivity around 458 nanometers. That is blue. It is shifted toward shorter wavelengths compared with sharks that live in shallow water. And the reason that matters is that blue is the only part of the visible spectrum that survives at depth. Red light is stripped out of seawater within the first few meters. Orange and yellow follow. What is left in the deep, and what almost every biological light source in the deep ocean produces, is blue. That is not the eye of an animal that gave up. That is an eye that was tuned, which means the parasite story gets stranger rather than simpler. The shark is maintaining a genuinely sophisticated low-light visual system, and then spending its life with something anchored to the front of it. So what is that eye actually for? Almost certainly not for spotting a seal at distance. There is no distance in the dark. The most defensible reading is that the eye exists for light that is made rather than borrowed. The flashes and glows that living things produce down there. Bioluminescence is everywhere in the deep ocean, and it is overwhelmingly blue. And a rod system peaking at 458 nanometers is aimed directly at it. A cloud of disturbed plankton lighting up as something swims through it. A flash from a startled squid. A glow that reveals where prey is, or where something else is hunting. You would not need sharp vision for that. You would need sensitivity, and a scratched cornea diffuses light far more than it blocks it, which degrades detail badly, but does much less damage to your ability to notice that a patch of the dark just lit up. There is a more theatrical idea in circulation, and I will give it to you with the label attached. Some have suggested that the trailing body of the parasite could serve as a lure, that it might attract prey close to the shark's mouth, turning an infection into an accidental fishing rod, which would make the whole arrangement a bizarre bargain rather than a parasitism. There is no solid evidence for that. It has not been demonstrated that this copepod produces light, and the lure idea remains a hypothesis that people find appealing partly because it is a great story. Treat it as unproven. The plainer explanation, that this is a parasite doing what parasites do, and the shark tolerates it because it can, fits the evidence better and is, in its own way, more disturbing. Because tolerating it is the real point. Think about what it takes to shrug off having your eyes colonized. Most animals cannot. An infection that degrades a primary sense is usually a death sentence delivered slowly. This shark has built a life where that is survivable, and it did it by not depending on the sense in the first place. So what is it depending on? Smell, first, and at the longest range. Currents carry chemical plumes for enormous distances in the deep. And unlike light, a smell does not need a straight line or a clear medium. It just needs time. And this is an animal with an unlimited supply of that. A carcass on the seafloor releases a plume that spreads for kilometers, and a shark that can travel patiently along a gradient, sampling and correcting for hours or days, will arrive. It does not have to be fast. It has to be right eventually. There is a strong case that this is how a great deal of feeding is initiated. Then, closer in, water movement. The same lateral line system the seals are using, running in the other direction. Sensors set into canals along the flanks and head, reading the near-field motion of anything disturbing the water. That is how you notice something struggling, or breathing, or swimming badly from meters away, in total darkness. And then at the last stage, the sense that makes this animal genuinely difficult to hide from. Sharks are electroreceptive. Their heads are studded with pores leading to gel-filled canals called the ampullae of Lorenzini, and these detect electric fields at extraordinarily low intensities. That matters because living tissue leaks electricity. A beating heart generates a field. So does a contracting muscle and a firing nerve. And the simple ionic difference between an animal's body fluids and the seawater around it. A living animal is a faint electrical object whether it wants to be or not. Electroreception does not need light. It does not need clear water. It does not care in the slightest what condition the cornea is in. Now stack those senses in order and you have the entire hunt without a single moment that requires speed or sight. Odor gets the shark into the right cubic kilometer of ocean. Water movement narrows it to the right few meters. Electroreception locks onto the exact position of the target in absolute darkness, at contact range, with enough precision to place a bite. The suction pulls, the upper teeth anchor, the body rolls, and the lower blade takes its plug. There is a detail buried in that sequence that quietly reopens the earlier argument, and it is worth noticing. Electroreception works best on something alive. A dead animal on the seafloor is a smell. Its muscles are not contracting, its heart is not beating, and the electrical signature that a shark's ampullae are built to detect fades as the body's chemistry breaks down. But a sleeping seal is a warm, wet, electrically noisy object hanging in the water column, broadcasting a field it has no way to switch off. That does not prove these sharks hunt. It does mean that the most refined short-range sense in their toolkit is one that would be largely wasted on a scavenger. So this is not a broken animal. It is a body that has been ruthlessly edited. Pay for what the environment rewards, refuse to pay for anything else, and accept damage in the systems that do not matter. Skeleton, minimal. Speed, abandoned. Corneas, surrendered to a tenant. And once you see the pattern, you can predict where else it shows up. Because there is one more system in this shark that has been pushed to an extreme, and it is the reason that a piece of this animal, straight out of the water, can put a person in the hospital. Cut a steak off a freshly landed Greenland shark, cook it properly, and eat it, and you will not simply feel unwell. Accounts of what happens describe something closer to poisoning that mimics severe drunkenness, loss of coordination, disorientation. a body that stops obeying instructions. The traditional Icelandic warning attached to this animal is not about the taste, and the compound doing it is not a venom, not a toxin the shark manufactures for defense, and not something it picked up from polluted water. It is the substance holding the shark's own proteins together. To understand why, you have to start with a problem that every fish in the ocean has to solve, and that most people have never thought about for a second. Seawater is saltier than the inside of an animal. Water moves across biological membranes from where dissolved substances are scarce toward where they are concentrated, and it does this relentlessly without needing energy because it is just molecules distributing themselves. So a fish sitting in the sea is a bag of relatively dilute fluid surrounded by a much more concentrated ocean, and physics is constantly pulling water out of it. An ordinary bony fish, a cod, a herring, a tuna, is being slowly dehydrated by the ocean it lives in. Its answer is brute force. It drinks seawater continuously, absorbs the water, and then spends energy pumping the excess salt back out through specialized cells in the gills. It is running a desalination plant in its own head every hour of its life, forever. Sharks and rays went a completely different way, and it is one of the most elegant solutions in vertebrate physiology. Instead of fighting the gradient, they erased it. A shark deliberately retains urea, the nitrogen waste compound that your kidneys work hard to get rid of, and holds it in its blood and tissues at concentrations that would be lethal in a mammal. That raises the total dissolved concentration of the shark's body fluids until it matches the surrounding seawater, or slightly exceeds it. And once you have done that, the water stops being pulled out. If anything, it drifts gently inward, which is a much easier problem to manage. The shark stopped bailing. It changed the water level. But you do not get a free solution in biology. You get a trade, and this one is severe. Urea is a protein denaturant. That is not a metaphor. It is what urea is used for in laboratories. If a biochemist wants to unfold a protein to take a precisely folded three-dimensional structure and reduce it to a floppy chain, one of the standard tools is a strong urea solution. It works by interfering with the delicate web of weak interactions that hold a folded protein in shape. Every enzyme in your body is a shape. Every one of them works because a particular pocket on its surface fits a particular molecule. Deform the shape, and the function is gone. So a shark that has flooded its own tissues with urea has, in principle, filled itself with a substance that should be quietly dismantling its own machinery from the inside. It does not, and the reason is the second half of the trick. Alongside the urea, sharks carry a counteracting compound, trimethylamine N-oxide, usually shortened to TMAO. TMAO is a stabilizer. Where urea pushes proteins toward unfolding, TMAO pushes them back toward their compact, correctly folded state. Hold the two in the right proportion. Elasmobranchs tend to run somewhere around 2 parts urea to 1 part TMAO. And the destabilizing and stabilizing effects cancel. The osmotic problem stays solved. The proteins stay folded. It is a chemical standoff maintained inside every cell, continuously, for the entire life of the animal. Now add depth, because TMAO does something else. Pressure is not a gentle force. At 2,000 meters, the water column above is pressing down at roughly 200 times the pressure at the surface. That does not crush a fish. It has no air spaces to collapse, but it does something more insidious. It squeezes molecular structures. Proteins occupy a certain volume when folded, and a different volume when unfolded or when bound to something. And pressure always favors whichever state takes up less space. So high pressure quietly shifts the balance of every reaction in the body, destabilizes some proteins, and can slow or distort enzyme function. TMAO counteracts that too. It is one of the main reasons deep-living animals can operate at depths that would derange the biochemistry of a surface species. And here is the pattern that makes this compelling rather than merely technical. Across many marine species, TMAO concentration in the tissues rises with the depth the animal lives at. Deeper animals carry more of it. It reads almost like a dial, turned further as the pressure increases. So now count what the Greenland shark has stacked on top of itself. It is a shark, so it is already running the urea strategy at full strength. It lives deep, so it needs high TMAO to keep its proteins functional under pressure. It lives cold, and cold has its own destabilizing effects on protein folding and on the fluidity of cell membranes. And it does all of this for centuries, in tissues that turn over slowly, in an animal that grows less than a centimeter a year. The result is flesh that is chemically saturated, not contaminated, saturated by design, with the compounds that are the only reason it works at all. That is why eating it fresh is dangerous. TMAO by itself is not the problem. The problem is what happens to it. Bacteria, including the ones already living in a gut, and the ones that get to work on the meat after death, carry enzymes that strip the oxygen off TMAO and convert it into trimethylamine. TMA is a small volatile amine. It is, incidentally, most of what you are smelling when you smell old fish, and in the quantities present in a several hundred-year-old deep-water shark, the effects reported are neurological rather than merely unpleasant. Meanwhile, the urea is doing its own conversion, broken down into ammonia by bacterial enzymes, which is why the meat's smell is so aggressive, that people describe it in terms of cleaning products rather than food. So the shark is not poisonous the way a stonefish is poisonous. Nothing about this evolved to hurt anything. It is a structural problem. This animal has built its entire biochemistry out of a compound that becomes a neurotoxic breakdown product the moment ordinary decomposition gets hold of it. And it did that because the alternative was not being able to live in the deep cold at all, which makes the human response to it one of the strangest food traditions on Earth. In Iceland, the meat is not discarded. It is processed. The traditional method is to bury or press the flesh so it drains and ferments over weeks, then hang it to dry in an open shed for months, exposed to the air, until what comes out the other side is a firm, pale, ammonia-heavy product that is eaten in small cubes and has become a piece of national identity and a reliable dare for tourists. Look at what that process actually is. It is the same reaction that would happen inside a person. Bacteria convert the TMAO and break down the urea. The difference is that the conversion happens outside a human body, in a shed, over months, and the volatile products vent into the air instead of into a bloodstream. Chemical analysis of the process has found TMAO dropping to undetectable levels within a matter of weeks of fermentation. Somebody, at some point, without any of that vocabulary, worked out that if you let the rot happen first, the meat becomes safe. That is not a recipe. That is a population figuring out a biochemical detoxification protocol by trial, error, and presumably a considerable number of very bad afternoons. There is a second consequence of this chemistry that is easy to miss and matters enormously later. Both urea and TMAO are less dense than an equivalent amount of dissolved salt, so a shark carrying its osmotic load this way gets a small buoyancy benefit for free. And this species stacks another, much larger one on top of it, an enormous liver, loaded with low-density oils, which in large sharks can account for a substantial share of total body weight. Sharks have no swim bladder. A bony fish holds depth by adjusting gas in a sealed sac, which is fast, precise, and a serious liability if you move vertically in a hurry. Sharks instead float on fat, Oil is lighter than seawater, so a big enough oil reservoir offsets the weight of the body and brings the animal close to neutral. For an animal built around not spending energy, that liver is not a fuel tank. It is the reason it does not have to swim to avoid sinking. Combine near-neutral buoyancy with almost no acceleration, and you get a body that can hang in the water column or drift over the bottom at close to zero cost. Barely more expensive to operate than the water around it. And it works. That is the uncomfortable part. Judged as an individual organism, this is one of the most durable vertebrates that has ever existed. Cold does not bother it. Pressure does not bother it. Darkness does not bother it. Starvation between meals is close to irrelevant when your metabolic demand is this low. Parasites take up residence, and it carries on. It appears to maintain functional tissue over timescales, where most vertebrates have been dead for generations. Nothing in the Arctic can meaningfully threaten a full-grown one. So here's the question that should be bothering you, and it is the one that turns this from a story about a strange animal into a story with actual stakes. If the individual is this hard to kill, why is the species in trouble? Because it is. And the answer has nothing to do with the shark's body, and everything to do with the calendar it is forced to live on. The same radiocarbon work that gave us the lifespan gave us a second number, and it is the one that should actually frighten you. Age at sexual maturity for females, estimated at around 156 years, give or take a couple of decades. Read that again slowly because the brain does not process it on the first pass. There are Greenland sharks alive right now that are older than every person you have ever met, older than the electric light, older than the theory of evolution, and have never reproduced. Not because they failed, because they are not old enough yet. A female of this species does not appear to become capable of reproducing until she reaches roughly 4 meters. And at growth rates measured in fractions of a centimeter per year, getting to 4 meters is not a phase of life. It is a century and a half of continuous, uninterrupted survival. Every winter, every storm, every predator, every parasite, every hook, every net, every stretch of bad luck. For 150 years, before the animal contributes a single offspring to the next generation. Put a human frame on it. A female born around the time of the American Civil War would, if she were one of these sharks, be arriving at her first pregnancy right about now. She would have spent the invention of the telephone, 2 world wars, the entire history of powered flight, and the whole of the space age as a juvenile. That is not a life history. That is a bet placed on the world staying stable for longer than any human institution has ever lasted. And here is where it stops being a fun fact and starts being a structural problem, because generation time is not a piece of trivia. Generation time is the clock that governs how fast a population can respond to anything. Every population is a balance between individuals being added and individuals being removed. Kill more than are added, and it declines. That is true for cod and for elephants and for this shark. The difference is the speed of the correction. A cod population that gets hammered can, under the right conditions, start rebuilding within a handful of years, because the survivors mature fast and spawn enormous numbers of eggs. The system has a short feedback loop. It can bounce. Now build the same feedback loop out of an animal that needs a century and a half to reach its first reproduction. There is no bounce. There is barely a response at all. If you remove adults from a Greenland shark population today, the replacements are not coming from sharks that will mature in 5 years. They are coming from animals that are currently 40 centimeters long and will not reproduce until sometime around the year 2180. The population's ability to correct itself is not slow. It is, on any time scale, a human institution can operate on, absent. And we do not even know the rest of the reproductive picture. This is one of the most striking gaps in the biology of any large marine animal. We do not know how long a pregnancy lasts, not approximately, at all. Gestation in this species has never been established, and given everything else about its pace, estimates that run to years rather than months are not unreasonable. But they are estimates. We do not have a confident number for litter size. Published figures vary wildly depending on the source and the assumptions, from small litters of a handful of pups up to estimates in the hundreds for the largest females. That spread is not scientific disagreement about a well-studied animal. It is what a data gap looks like when people try to fill it. We know the pups are born live, nourished by a yolk sac rather than a placenta, and that they arrive at something under half a meter, a shark you could lift with one arm. We do not know where they are born. No confidently identified nursery ground. No documented pupping area. The newborns of the longest-lived vertebrate on the planet appear somewhere in the North Atlantic or the Arctic, and nobody can tell you where. Sit with the asymmetry there. We can date an individual of this species to within a century using nuclear fallout chemistry, and we cannot tell you how long its mother was pregnant or where she went to give birth, which loops back to something established earlier and gives it a much darker meaning. Remember the result from the eye lenses. The bomb pulse signal only showed up in the smallest sharks in the study, roughly the ones 220 centimeters and under. That was presented as a dating breakthrough, and it is. But turn it around and look at what it says about size. A Greenland shark over 2 meters long is a child. An animal longer than the person holding it, heavy enough to need a winch, with a full set of those interlocking cutting teeth, is decades away from maturity at best, possibly a century away. There is no stage in this animal's life where being large means being an adult in the way we instinctively assume. The intuition that a big fish is a mature fish, which underpins an enormous amount of how people think about fishing, is simply false here. So every small Greenland shark that comes up in a net is not a spare. It is the next century. Now put the numbers together and watch what happens to any idea of recovery. Suppose a population is reduced. To rebuild, the surviving juveniles have to reach maturity. Call it a century and a half. Then their offspring have to do the same. 2 generations of this animal is roughly 300 years. That is longer than the entire industrial era. It is longer than most countries have existed in their current form. A conservation plan for this species, if it worked perfectly starting today, would produce its first genuinely meaningful result long after everyone who wrote it, funded it, enforced it, and argued about it is dead, and probably after the institution that created it has been dissolved and replaced twice. We are not built to manage anything on that timescale. Our fisheries agreements are renegotiated every few years. Our political attention spans are measured in months. The animal we are talking about does not register a human policy cycle as an event. The formal assessments reflect this in a way that is quietly extraordinary. When the species was reassessed and moved to vulnerable on the IUCN Red List in 2020, the population decline was evaluated over a window of roughly 420 years because the standard method looks back across 3-generation lengths. And 3-generation lengths for this animal reaches back past the founding of most modern nations. The estimated decline over that window is on the order of 60%. Think about what it means that the appropriate assessment window for a living species is 4 centuries. The baseline is not a memory. It is not even a record. It is a reconstruction of an ocean that no living person could have seen. And the pressure has not stopped. It has only changed shape. Estimates cited by the IUCN put accidental catch at something on the order of 3,500 individuals a year across the Northwest Atlantic, the Arctic Ocean, and the Barents Sea, taken in bottom trawls, on long lines, in gill nets. Gear set for halibut, gear set for shrimp, gear set for cod. Nets and lines dragged through cold, deep water for entirely different reasons, which is exactly the water these sharks occupy. None of that is malice. Almost none of it is even intentional. Nobody is fishing for Greenland shark anymore in any serious commercial way, because the meat is the chemistry problem we just walked through, and there is no market worth the trouble. That is what makes it so grim. The species is not being hunted. It is being deleted as a side effect. Every year, several thousand animals that took anywhere from decades to several centuries to build come up on someone else's gear, and a proportion of them do not go back alive. And even the ones released have been hauled from depth, handled, and dropped back into water they now have to survive in, with whatever damage they took on the way up. 3,500 a year does not sound like a catastrophic number. For a cod population, it would be nothing. For an animal that adds a new breeding adult roughly once every century and a half, per individual, it is a subtraction the population cannot answer. Some protection has arrived. Fisheries bodies in the North Atlantic have moved in recent years to require release of Greenland sharks taken as bycatch, which is genuinely meaningful and long overdue. Whether it is enough is not something anyone can evaluate for a very long time, and that is the recurring problem with this animal. The feedback that would tell you whether you got it right arrives on a timescale nobody can wait out. But none of this is the first wave, because before the accidental catch, before the trawlers, before anyone had the faintest idea what these animals were or how long they had been alive, there was a period when people went looking for them on purpose, deliberately, in enormous numbers, and they were not doing it for the meat, which was worthless, or for the fins, or for sport. They were doing it for the one part of the animal that was worth real money, the part that had been keeping it afloat. A large shark's liver is not a small organ. In a big Greenland shark, it is a vast, soft, oil-heavy mass filling a huge fraction of the body cavity, and it exists to do the job a swim bladder does in a bony fish: make the animal light enough to hang in the water without working for it. That oil is also an excellent industrial product. It burns cleanly in a lamp. It stays fluid when it is cold, which mattered enormously in a world of unheated workshops and machinery running through northern winters. It lubricates. It treats leather. Before petroleum distillation became cheap and universal, oil from marine animals was one of the primary ways the industrialized world lit its rooms and kept its machines from seizing, and the North Atlantic was hunted comprehensively for it. So people went out and got it. The method did not require technology. It required a line, a hook, bait, and patience. You could take these sharks from a small boat or from a hole cut through the sea ice, hand-hauling them up out of the cold, and the operation was reliable in a way that shark fishing usually is not. And the reason it was reliable is the part that turns the stomach a little. This animal does not fight. Muscles running at the temperature of near-freezing water cannot produce the violent, sustained struggle that makes hooking a large shark dangerous and difficult. A hooked mako is a catastrophe on a line. A hooked Greenland shark is a weight. The exact set of adaptations that let it survive for centuries is the set that made it trivially easy to kill. That is not irony. It is a straightforward consequence. Endurance strategies work beautifully against cold, hunger, pressure, darkness, and time. They have no answer at all for a hook. Now the scale, and this is where the story stops being about individual animals. Estimates for the early 20th century put annual catches across Greenland and Norway somewhere in the range of tens of thousands to well over 100,000 sharks per year. In Norway, the peak year on record is 1948, with roughly 2.8 million kilograms of shark liver oil landed, a figure that has been translated to something in the region of 58,000 individual animals in 1 year, from one country's fleet. Now do the arithmetic that nobody could have done at the time, and treat it as an illustration rather than a measurement, because we do not have the age structure of that catch and never will. If the average animal taken in that season had been alive for even a century, a conservative figure given that a 2-meter juvenile is already decades old, and the animals worth hauling were the big ones, then a single year of that fishery removed something on the order of 5 to 6 million accumulated shark years from the North Atlantic. 5 million years of life, landed in 12 months, rendered into oil, burned. And the shape of the fishery over time tells its own story. The same story that plays out everywhere a slow-growing animal meets an efficient industry. In the 17 and 1800s, Norwegian fishermen took Greenland sharks close to shore, near the coast, without much effort. By the early 1900s, they were going further out, then further, then across to Greenland waters to keep the supply up. That progression is not a story about improving boats. Fleets do not sail progressively further from home for fun. They do it because the animals near home are gone. The expanding search radius is the fingerprint of local depletion, written in the logistics. Then the fishery ended, and it is worth being clear about why, because it was not a rescue. Petroleum won. Synthetic lubricants won. Once you could crack oil out of the ground and refine it to whatever specification you wanted, killing a several hundred-year-old animal to fill a lamp stopped making commercial sense, and the directed fishery collapsed and had largely ended by around 1960. The Greenland shark was not saved. It was made obsolete. It survived because it stopped being worth money, which is a very different thing from being protected, and it is a distinction worth holding on to, because it means nothing about that outcome was a decision. If synthetic lubricants had arrived 50 years later, we would be having a different conversation, and possibly not having one at all. But the detail that genuinely lands is the timing of what we knew. The directed fishery ran through the 19th century and peaked in the middle of the 20th. The radiocarbon eye lens work that told us what these animals actually are came out in 2016, more than half a century after we stopped killing them in bulk. We found out what we had been killing. Every person who ever hauled one of these sharks over a gunnel believed they were pulling up a large, ugly, sluggish, commercially useful fish. There was no framework in which to understand that the animal on the deck might have been swimming when their great-great-great-grandparents were born. That information did not exist. It could not have existed. The method that produced it depended on a nuclear signature that had not been created yet, applied to a tissue nobody had thought to look at, which means an entire industry operated for generations on an animal whose most important biological property was invisible to everyone involved. That has a consequence beyond guilt, and it is one that fisheries science understands very well now. When you fish a population hard, you do not remove individuals evenly. You remove the big ones first because the big ones are the ones worth taking, and in a species like this, the big ones are the only ones that reproduce at all. What is left behind is a population with its age structure truncated, plenty of juveniles, a hollowed-out middle, and the century-and-a-half-old breeding females stripped out, in fast-growing species that damage repairs itself in a decade or two. In this one, the repair has to be manufactured out of animals that will not be ready for another century. The North Atlantic that existed before that fishery, with its full complement of 4 and 5 meter animals that had been accumulating since the 1600s, is not something anyone alive has seen, and it is not something anyone alive is going to see. That baseline is gone in the most literal sense available. Rebuilding it requires more time than the entire history of industrial fishing. So the 60% decline estimated across 4 centuries is not an abstraction. It has a mechanism, and the mechanism is a lamp. And this is the point where you would expect the story to settle into something manageable. A cold-water Arctic species, historically overfished, now slowly persisting in the far north where we can at least draw a box around it on a map and know where it is. Except we cannot. Because in 2022, one of these animals turned up somewhere it had absolutely no business being. Glover's Reef sits off the coast of Belize. It is a coral atoll in the Western Caribbean. Turquoise shallows, reef crest, tropical sun, the water at the surface warm enough to swim in all day. A research team was working there on tiger sharks, with Belizean fishermen who fish the deep slope where the atoll drops away. They set gear down the outside of that slope, into the cold water below the reef, and hauled up something nobody in the Western Caribbean had ever recorded. A sleeper shark, around 3 to 3.5 meters. The blunt head, the small eye, the unmistakable body plan of a somniosis. The account of what happened next is almost comically frustrating and entirely believable. A storm was coming in. The animal had to be released quickly, for its own safety and everyone else's, and there was no time to take a genetic sample. Photographs, measurements, and then it was gone back down the slope. Which means the identification is exactly as certain as it can be, and no more. The published report in Marine Biology in 2022 describes it as most likely a Greenland shark, with the possibility that it was a hybrid between the Greenland shark and its Pacific relative, the Pacific sleeper shark. Without tissue, that cannot be resolved. It never will be for that individual. But even at the loosest reading, a sleeper shark was hanging off a tropical coral atoll, thousands of kilometers from the nearest sea ice. And the first instinct is to call it a wanderer, a lost animal, something that took a wrong turn. That instinct is wrong, and correcting it is the single biggest reframe in this entire story, because the ocean is not organized the way a map suggests. We draw ranges as colored blobs on a flat picture of the world, and that picture is a lie of dimension. It shows latitude and longitude and completely hides the axis that actually matters to a deep water animal. The ocean is not a surface. It is a volume, averaging around 4 kilometers deep, and the property that governs where a cold-adapted animal can live is not how far north it is temperature. And temperature in the ocean is stratified. Sunlight heats the top. Wind mixes that heat down a little way, and then it stops, because warm water is less dense than cold water and floats on top of it. Below that mixed layer, there is a zone where the temperature falls sharply, the thermocline. And below the thermocline, the ocean is cold everywhere. Not cold at the poles, cold everywhere. Go down to 2,000 or 3,000 meters beneath the equator, in water where the surface is 28 degrees, and there are flying fish and coral, and the temperature is a few degrees above freezing. It is dark, it is under enormous pressure, and it is very nearly as cold as the water off Svalbard. That deep cold layer is not a regional feature. It is the majority of the ocean by volume, a single continuous mass of near-freezing water wrapping the entire planet, and the surface warmth we experience is a thin skin floating on top of it. Now put a Greenland shark into that picture properly. This is an animal with a temperature envelope running from just below 0 to somewhere around 5 or 6 degrees. That envelope does not describe the Arctic. It describes the deep ocean. The Arctic is simply the place where the deep ocean comes up to the surface. That is what the polar regions actually are, physically. They are where the cold layer outcrops, where the water that is normally 2 kilometers down under the tropics is available at the top, exposed to the sky and the ice, which means that from a Greenland shark's perspective, the Arctic is not home in any special sense. It is a convenient shallow shelf where its preferred water happens to be lying against the surface. Everywhere else, that same water is still there. It is just underneath something. So a sleeper shark on the deep slope of a Caribbean atoll is not lost. It is in exactly the right water. It is in the same water it would occupy off Greenland. The only thing that has changed is that there are several hundred meters of warm tropics stacked on top of it, and a reef with parrotfish on it somewhere far above. And the moment you accept that, the map falls apart. Because if these animals can occupy the cold layer at depth, then the reason we mostly find them in the north is not necessarily that they mostly live in the north. It is that in the north, their water is shallow enough that our gear reaches it. We fish the Arctic and subarctic shelves intensively, at depths where a Greenland shark is catchable, and so that is where Greenland sharks are caught. In the tropics, the same water is a kilometer or more down, largely below the depths where commercial fishing happens and far below where anyone is looking. The distribution map is not a map of the animal, it is a map of us. That is the same failure that opened this story, and it is worth naming, because it keeps happening with this species. We could not age these sharks and concluded they were unmeasurable, when the truth was that our aging methods only work on animals that mineralize. We assumed a slow shark could not be a predator, when the truth was that our idea of predation was built around burst speed. Now we assume an Arctic species, when the truth may be that we have been sampling the one narrow window where its habitat happens to be within reach. Every single time, the limit turned out to be ours. Two things need to stay honest here, though, and both of them matter. The first is that one record is one record. A single animal on the slope of one atoll, without a genetic sample, does not establish a Caribbean population. It does not tell you how many there are, whether they breed there, whether they are permanent residents or occasional deep travelers moving along the cold layer. It widens the question. It does not answer it. Anyone who turns that single catch into Greenland sharks live in the Caribbean is running past the evidence. The second is that this idea was not invented by that catch. Records of sleeper sharks at depth in warm regions had already been accumulating from the Gulf of Mexico and elsewhere, which is why the report was framed as further support for something people already suspected rather than a bolt from nowhere. The Belize animal did not create the hypothesis, it hardened it. And there is a much larger pattern sitting behind it, which is the behavior of the whole group these sharks belong to. The Greenland shark is not alone. It has close relatives in the same genus. And where they turn up is the argument, made independently, by animals nobody was trying to prove a point with. In the North Pacific, there is the Pacific sleeper shark. A near-identical body plan, blunt, slow, enormous, occupying the cold water from the Bering Sea and the Chukchi down the coast of North America, which fits comfortably with the idea of a cold northern animal, until you follow the records. It has been documented far outside that comfortable picture, in the tropical Pacific, and into the South China Sea, and its recorded depth range runs from the surface down past 2,000 meters. In the southern hemisphere, there is another one, the southern sleeper shark, described from the far south of South America, off South Africa, around southern Australia and Tasmania and New Zealand, and out into the southern Indian Ocean, living down the continental slope in the hundreds of meters. Whether it is genuinely a separate species is itself contested. Molecular work has raised the question of whether it should be split off at all, which is its own small reminder of how thin our grip on this group is. Now step back and look at where that genus is. Top of the Atlantic, top of the Pacific, bottom of the world, and, in the records that keep accumulating, in warm latitude waters at depth in between. That is not a distribution of polar animals with a few strays. That is the map of the cold layer. These sharks are found at the surface, where the cold water is at the surface, and they are found a kilometer or two down where the cold water is a kilometer or two down. And the only reason we filed them as polar species in the first place is that the poles are where we could reach them. And notice how neatly it fits with something we already established. The rhodopsin in this animal's rods peaks in the blue, shifted away from what shallow water sharks carry. That is a deep water eye. It is the eye of a species whose evolutionary history has been spent in a light environment where only the blue survives, which is to say, well below the surface, not the eye of an animal that has adapted to a dark Arctic winter at shallow depth. The visual system was telling us where this species really lives before anyone caught one under a coral atoll, which leaves an unpleasant practical problem sitting in the middle of everything we said about conservation. How do you assess a population when you do not know its boundaries? Every population assessment depends on defining what you are counting. Fisheries bodies estimating decline, setting bycatch measures, deciding whether a species is vulnerable, all of it assumes you have some grip on the size and shape of the thing you are managing. If a meaningful part of a species is sitting in cold, deep water at latitudes nobody surveys for it, then the numbers are being calculated on the visible fraction, and nobody can tell you what fraction that is. That cuts both ways, and I am not going to pretend it only cuts the frightening direction. It is possible there are more of these animals than we think, distributed through deep cold water across a far larger area than the maps show. That would be good news, and it is a real possibility. It is also possible that the deep tropical animals are sparse strays from the same northern populations that were stripped for lamp oil, in which case there is no hidden reserve and the assessment is, if anything, generous. Nobody knows. And the reason nobody knows leads directly into the hardest thing about this species, which is not its biology at all. It is that almost everything we have said about it comes from animals that were already dead. You cannot take the lens out of the eye of a living shark. That single sentence contains most of the reason this animal remains a mystery. The method that finally cracked the age question is destructive. It requires the animal to be dead already, which in practice means it requires the animal to have been killed by somebody else, in a fishery that was not looking for it and then made available to science. So the entire body of knowledge about how old these things get is built out of accidents. The same is true for almost everything else. Stomach contents require opening the animal. Reproductive anatomy requires dissection. Tissue chemistry, retinal structure, the composition of the eye, all of it comes from specimens, and specimens come from mortality. That builds a specific and unavoidable set of biases into the picture, and it is worth naming them plainly, because they are the reason so many statements about this species have to be hedged. The specimens come from where fisheries operate, which is a narrow band of the ocean skewed toward continental shelves at northern latitudes and toward depths that commercial gear reaches. They come in the sizes that particular gear catches, which is not a random sample of the population. And the study that produced the famous ages worked with 28 animals, all female. 28, for the entire established lifespan of the longest-lived vertebrate known. That is not a criticism of the work. It is remarkable that the work exists at all, given what it takes to get material like that. It is a description of how thin the evidence necessarily is. When you hear a number attached to this species, the sample behind it is usually small enough to fit in a room. So why not just watch them? Because watching them is close to impossible, and the reasons are the same reasons the animal is interesting. They live in cold, deep, dark water. Large parts of their range are under ice for much of the year. Deep water observation is expensive. Remotely operated vehicles need a ship, a crew, a cable, weather, and a budget, and every hour of footage costs real money. Baited cameras dropped to the bottom are cheaper and have produced genuine results, including the first attempts at estimating local abundance in Arctic waters, but a baited camera shows you one small circle of illuminated seafloor, and whatever chooses to enter it, and when a Greenland shark does enter it, the footage is strange in a way people do not expect. It arrives without any drama. Most large predators announce themselves. You see the approach, the turn, the interest, the acceleration. This one appears at the edge of the light like something being slid into frame. It does not startle. It does not flinch from the lamps. It does not do the nervous, high-speed circling that other sharks do around bait. It moves in, occupies the space, feeds if it feels like it, and then leaves the way it came. And the whole encounter has the tempo of watching a large object drift. That non-reaction is itself informative. This is an animal that appears to have no meaningful predator and no reason to be alarmed, in an environment where fleeing is a waste of energy. But it also means the footage tells you very little. Almost nothing happens on a timescale a camera can capture, and that is the real wall, the one that no amount of funding solves. Suppose you could follow a single Greenland shark continuously every second with perfect coverage. Suppose you watched it for a solid year. An absurd, unprecedented commitment. More continuous observation than has ever been achieved for any large marine animal. You would have covered less than 0.5% of its life. To watch one of these animals live a full life, from birth to death, you would have to observe without interruption for something like 3 centuries. You would need to start before the invention of the telephone and finish sometime in the future, and hand the equipment down through 10 generations of researchers. Now flip that around and look at what it does to the science. Marine biology, as an organized discipline, is younger than a great many of the individual sharks it is studying. The field did not exist in a recognizable form when some of these animals were born. There are Greenland sharks in the North Atlantic right now that are older than every scientific institution that has ever studied them. Every single thing we know about this species is a snapshot. We have never watched one grow up. We have never watched one mate. We have never seen one give birth or found the place where it happens. We have never followed an individual across even a small fraction of its life. What we have instead is a scattering of dead animals from different sizes and places, and a set of inferences stitched between them, which is why the honest version of this story is full of phrases like estimated, not established, and remains unknown. We are trying to reconstruct a 300-year life from a few hundred still frames taken by strangers who were fishing for something else. And that gap between the animal's timescale and ours is not just an academic problem. It is about to become a practical one, because the one part of the ocean where these sharks are accessible to us is the part changing fastest. Remember what the Arctic actually is in this story. It is the place where the deep cold layer outcrops at the surface. It is the shallow window into a habitat that is otherwise kilometers down. That window is warming faster than almost anywhere else on the planet, and the sea ice that has covered it is retreating. Follow what that does, step by step, without any speculation about the sharks themselves. Less ice means more open water for more of the year. More open water means longer operating seasons for fishing fleets and access to grounds that were previously closed by ice for much of the year. Northern fisheries are expanding into areas that were not commercially workable a few decades ago. Shipping routes are opening across water that used to be solid. Every one of those developments puts more gear, for more months, into exactly the depth band and exactly the water where Greenland sharks are catchable. The bycatch pressure that already removes thousands of these animals a year is not a fixed quantity. It is a function of how much fishing effort overlaps their habitat, and that overlap is increasing for reasons that have nothing to do with sharks and everything to do with ice. Then there is the water itself. This is a species with a temperature ceiling somewhere around 5 or 6 degrees. If the shallow Arctic layer warms past that, the shark's response is straightforward. It goes deeper, into water that is still cold, because there is always colder water below. Physiologically, that costs it very little. Ecologically, it might cost it a great deal. And this is where I want to be careful, because it is a plausible consequence rather than a documented one. The reason the shallow Arctic matters to these animals is not the temperature. It is the food. That is where seals rest. That is where carcasses from the surface world fall in. That is where the shallow Traumeo shelf concentrates the productivity of a short, violent Arctic summer into something a scavenger can find. A shark pushed down and away from that shelf is still in comfortable water, but it is further from the falling remains of everything that dies at the surface. Meanwhile, the Arctic food web above it is being rearranged as subarctic species push north into warming water, and ice-dependent species lose the platform they hunt and breed on. The specific animals that end up as carcasses on the bottom, and how many of them, and when in the year, are all being reshuffled. An animal with a metabolism this low can wait out an enormous amount of scarcity. Waiting is what it is built for, but an animal that needs a century and a half to produce its first offspring has no capacity to adapt to a changing system in the way a fast-breeding species can. Adaptation by natural selection requires generations, and generations here are longer than the entire period over which the Arctic has been industrializing, so it cannot evolve its way out on any relevant timescale. It can only endure or not. And here is where all of it converges into something genuinely difficult to hold in your head. There are individual animals in that water right now that were born before the fishery, before the industrial use of fossil fuels at scale, before the thing that is currently warming their habitat had been invented. A shark that started life in the mid-18th century has spent its life in a North Atlantic that was quietly reorganized around it. Hunted, then abandoned, then reopened, iced, then thawed, emptied of whales, refilled, emptied of cod, restocked with gear. It did not experience any of that as history. It experienced it as water. So let us build one. Not a specific animal. Nobody has a specific animal with a birthday, and anyone claiming otherwise is inventing precision. Let us build the most conservative possible individual the published evidence supports, and then follow it, take the floor of the range. Not the headline figure of nearly 4 centuries, not the upper end of the error bar, the minimum. A lifespan of around 272 years, which is the least dramatic number the radiocarbon work will allow. Run that backward from today, and you land somewhere in the mid-18th century. The animal is born at roughly 40 centimeters, small enough to hold in your hands, in cold water somewhere nobody has ever identified. It has no parental care. It has no nursery ground that anyone has found. It has a functioning set of those interlocking lower teeth and a rod-packed eye tuned to blue light. And from the first day, it is doing the only thing this species does, existing at the lowest possible cost. And it grows, faster now than it ever will again, which is not saying much. Even the quickest stretch of this animal's life is a couple of centimeters in a year, and the pace only falls off from there. Above it, the North Atlantic is filling with sails. The great age of whaling is running through the 18th and 19th centuries, and it does something to the deep ocean that took 200 years for anyone to think about. A dead whale that sinks is the single richest food event in the deep sea. Tons of fat and protein arriving on a seafloor, where food normally drifts down as a slow snow of particles. Whale falls support entire specialized communities for years. And industrial whaling removed an enormous fraction of the largest animals in the ocean over the course of a couple of centuries. What that did to deep-sea scavengers over the long run is a genuine, open question, not a settled one. In the short term, whaling may have increased the supply of carcasses and stripped carcasses hitting the water. Over the longer run, taking that much large animal biomass out of the system had to reduce the standing supply of whale falls. Either way, a slow shark that eats what sinks was living through a 2-century reorganization of its own food supply, and it had no way to register the change as anything other than better or worse years. Meanwhile, it is still growing. It passes a meter. It passes 2. The size at which anyone hauling it up would call it a full-grown shark and be wrong by more than a century. It is not reproducing. It will not reproduce for a very long time. By the time it crosses 3 meters, there are steamships crossing overhead. By the time it approaches 4, the size where a female of this species is thought to become capable of breeding, the calendar has rolled into the 20th century, which is exactly when the liver oil fleets were at their most efficient. That is the part of this hypothetical that is genuinely hard to sit with. If our conservative animal is female, if Farhanasine, she spent roughly a century and a half getting to the point where she could contribute anything at all to her species, and she arrived at that point at the historical moment when tens of thousands of her kind were being pulled out of the water every year for lamp fuel and machine grease. She survived that by luck. Nothing about her body helped. Her body, as we established, is close to defenseless against a hook. Then the fishery collapses, not because anyone protected her, but because petroleum was cheaper. And then the bombs. This is where the story does something that I find genuinely eerie, because it stops being a metaphor and becomes an actual physical fact about the animal's tissue. The core of that shark's lens, the part built before it was born, in the 1700s, holds carbon from an ocean that had never seen a nuclear test, no bomb signal. That is precisely how we know how old it is. But the lens does not stop growing. New fiber cells are added to the outside of it throughout life, layer over layer, and each of those layers is built from the carbon available at the time it formed. So the outer layers of that same lens, laid down in the 1950s and 60s, are built from carbon that carried the fallout spike, which means the boundary of the nuclear age exists inside that animal's eye as a physical stratum. There is a line in there. Below it, an 18th century ocean. Above it, ours. The shark has been carrying the transition between those two worlds in its own tissue, in the correct order, in a structure that cannot rewrite itself. It is a core sample of the 20th century, walking around, alive, in the dark. And it does not know. That is not sentimentality. It is a statement about scale. Nothing in this animal's sensory or cognitive world registers centuries. It registers cold and pressure and chemical gradients and the electrical flicker of something living within a body length. It has experienced the entire industrial history of the human species as minor variations in what its water tastes like and what falls out of the sky above it. So what have we actually been looking at this whole time? Not a monster. Nothing about this animal is built for aggression. Its whole architecture is the opposite of aggression. It is refusal, and not a marvel either. Not in the way that word usually gets used. There is nothing miraculous in it. There is no secret ingredient, no fountain, no anomaly. Every single strange thing about this shark is a line item on the same bill. Run the ledger. It gave up a mineralized skeleton, and in exchange it got a body that costs less to carry and does not fight the water. The price was that it also gave up the only record of its own age and stayed unmeasurable for the entire history of modern biology. It gave up burst speed, and in exchange, it got a metabolic rate low enough to stretch its life across centuries. The price was that it can never chase anything and can never escape anything either, including a handline dropped through a hole in the ice. It gave up the clarity of its own eyes to a parasite, and in exchange, it saved whatever it would have cost to defend a sense that a lightless environment barely rewards. The price is that we will never be sure how much of the world it can actually see, or whether the tuned, blue-shifted retina behind that clouded cornea is being used for anything we have thought of. It flooded its tissues with urea and TMAO, and in exchange it got proteins that stay folded under 200 atmospheres in water below freezing. The price is flesh that turns neurotoxic the moment ordinary bacteria get to it, which made it commercially useless as food, which is the only reason the fishery targeted the liver instead, which is the only reason the animal was worth killing at all. And it pushed reproduction back a century and a half, which is what you do when you expect to have unlimited time. The price is a species that cannot respond to anything. Not to fishing, not to warming, not to a change in what sinks to the bottom. Its entire strategy is a bet that the world will stay roughly the same for 300 years at a stretch, and it has been winning that bet since long before anything on this planet was capable of breaking it. Every one of those trades makes sense on its own. Together, they produce an animal that is nearly impossible to kill by natural means and trivially easy to kill by accident, which is why the age is not really the story. We keep leading with the number because the number is astonishing, and it should be. But the number is not the point. The number is the receipt. 3 centuries is what shows up on the invoice when an animal spends almost nothing for 3 centuries. It is not an achievement the shark is working toward. It is the arithmetic left over after every other cost has been refused. And that changes what this animal is, in a way worth being precise about. We call it ancient. It is not ancient. A 400-year-old shark is not a survivor from a lost world any more than a 400-year-old tree is a survivor from the 17th century. It is a currently living organism that happens to have a long life. There is a habit of calling animals like this living fossils, as though they are leftovers that forgot to change. That framing is wrong here, and it obscures the actual point. This is not a primitive animal that got stuck. The blue-shifted rhodopsin is not primitive. The interlocking cutting blade in the lower jaw is not primitive. The osmolite chemistry tuned to pressure and cold is not primitive. This is a highly specialized modern species, precisely adapted to a habitat that happens to be cold, dark, high pressure, and enormous. It looks archaic to us because it is blunt and slow and unbothered, and we have been trained by warm, shallow water to read speed as sophistication. The strangeness is not in the shark. It is in our clock. We have no trouble with slow plants. Nobody finds a bristlecone pine disturbing. We accept that a tree can stand through the entire history of a civilization because trees do not compete with our idea of what an animal is. But a predator that outlasts nations breaks something because our entire mental model of predation was built out of things we can see. The strike, the chase, the burst, the kill. All of that comes from a thin, sunlit, oxygen-rich, temperature-forgiving film at the very top of the ocean, which is the only part of it we ever swim in. That film is a rounding error. The ocean averages around 4 kilometers deep. Below the first few hundred meters, it is dark, and below the thermocline, it is cold. Everywhere, permanently, at every latitude. By volume, most of the living space on this planet is a cold, black, high-pressure environment where food is scarce, movement is expensive, and nothing has any reason to hurry. Which means the Greenland shark is not an exception to how life works. Measured against the actual habitat available on Earth, an animal that is slow, cold, patient, chemically stabilized against pressure, indifferent to darkness, and built to last for centuries, is closer to normal than we are. We are the strange ones. Hot, fast, oxygen-hungry, burning through a lifespan in a few decades because we live in the one narrow layer where that is affordable. And that reframe leads directly to the last uncomfortable thought, which is the one to leave with. We only found out about this animal because of a coincidence. Not a discovery, a coincidence. It required a tissue that refuses to be rebuilt. A lens core sealed before birth because transparency forbids maintenance. It required a species of parasite-ridden, commercially abandoned shark to keep turning up as bycatch in someone else's nets. And it required a decade in the 20th century when humans detonated enough nuclear weapons in the open atmosphere to leave a chemical timestamp in every living cell on Earth. Remove any one of those 3 and we would still be guessing. There would be no proof. The claim that these animals live for centuries would still be a suspicion supported by a handful of tag recaptures and an extrapolated growth rate, which is exactly what it was until 2016. So the obvious question is, how many other animals are down there on a clock like this? Because most species do not have a convenient sealed tissue. Most invertebrates in the deep sea have nothing analogous to a lens nucleus. Most of them have never been aged at all by any method, and a good number have never been observed alive. The deep ocean is full of animals we know only from specimens hauled up dead and pickled. And the specific question of how long they live is one we have almost never been able to ask, let alone answer. We have already been wrong once in the most spectacular possible direction on one of the largest and most conspicuous animals in the Arctic. There is no particular reason to think it was the only one. So picture the last image, and let it be the accurate one. A remotely operated vehicle somewhere in the North Atlantic, a kilometer or more down. Its lights throw a cone into the water, and the cone ends. Not in darkness gradually, but in a wall. Because particles in the water scatter the beam, and beyond a few meters, there is nothing to see, and nothing to see it with. Inside the cone, marine snow drifts down through the frame, endlessly. The slow fall of everything that has died above. The temperature outside the housing is close to freezing. The pressure is around 100 times what you are sitting in, and it climbs with every meter below. And a shape comes into the light from the left, without any announcement, pale gray, blunt-headed, skin scarred in the way that it only accumulates over an unreasonable amount of time. Small eyes with something pale anchored to the front of them, trailing loose and drifting with the water. The body is enormous, and it is not doing anything. No reaction to the lights, no interest in the machine, no fear, no aggression. The tail sweeps once, then a long pause, longer than feels right, the animal simply gliding. And then it sweeps again, and the body slides out of the cone of light and is gone. And the water where it was is black. And it looks exactly the way it did before. That animal did not begin when the light found it. It has been moving through that water at that speed, in that dark, at that temperature, since before the country you are watching this from had a name. Through the whaling, through the lamps, through the bombs, whose fallout is sitting in its own eye, through every argument we have ever had about what it is. The light will move on in a minute. The ship will pull the cable up. The recording will end. And it will still be down there, going nowhere in particular, in no hurry at all, adding half a centimeter a year.